The central dogma comprises two fundamental processes, which is transcribing the DNA to form an RNA and then decoding the RNA to form a protein with amino acid sequence. So the sequence of the amino acid, that message has to be present in the DNA. And therefore, in this session, the story of the genetic code will unfold. Let us therefore look at what are the learning outcomes of this session. There is a linear correlation between the sequences of nucleotides in the DNA and the sequence of amino acids in the proteins. The genetic code, again, was cracked due to very simple experiments. The Wobble hypothesis and the codon degeneracy are two very important phenomena of the genetic code that allows changes in the nucleic acids, but it may not reflect in the proteins. And thereby, there is a minimization of the deleterious effects of mutations. Also interesting is the fact is that the start and the stop codons decide the reading frame of the protein in the DNA. So let us look at what are the different features of the genetic code. So there are three rules that govern the genetic code. Rule 1 is that the genetic code is read from 5' to 3'. So let us consider having a hexanucleotide or a 6-mer, and considering the fact that the 6-mer has a 5' end, and of course it will have therefore a 3' end. So this is the 5', this is a 3' end, and as per the rule, this has to be read in this direction. So what results after translation is a protein, or rather a dipeptide made up of threonine and arginine, indicating that ACG codes for the amino acid threonine and CGA codes for the amino acid arginine. Also, what is interesting to note over here is that one amino acid is coded by a codon that is a triplet. That means 3 nucleotides are forming a codon. Now, let us take another instance. So here, okay, this is what we meant. So if the codons are read in the 5' to 3' direction, the proteins are formed from the N terminus to the C terminus. Let us consider another case in point where we are just reversing the sequence. So you have ACG, okay, with CGA, okay. But how is it? It is that you have made this hexanucleotide 3' over here and 5' over here, which means that this is going to be read in this direction. So the dipeptide formed is now different because the codons that read from here could be coding for a different amino acid. Let's look at another case in point. Again a hexanucleated, but now what has been done is that the second codon has been kept first and the first codon has been kept second. So what only happens is that you again get a dipeptide, but then you have the arginine as the N-terminal amino acid and the threonine as the C-terminal amino acid to form a dipeptide again. So basically what we understand from all these three examples is that reading will happen only from the 5' to 3' direction. The second rule is that the genetic code is non-overlapping and contiguous. So let us look at what does that mean. So consider the same hexanucleotide, okay. So the first three codons, okay, the first three nucleotides in the codon codes for threonine as we already saw, and the second codes for arginine. Now you can see that this codon and this codon have independent identity of their own. There is no overlapping of any of the nucleotides. So it is not overlapping with each other, and also it is continuous, that is one after the other. Let us look at a scenario if the genetic code were not overlapping. But before that, we understand that here a dipeptide has been formed. Now, the same sequence, okay, but here we will consider that the codons are overlapping. So this becomes the first triplet to be read, and that stands for threonine. The second triplet overlaps with the first one. So it is continuous, but then the codons are overlapping. So the next codon in turn will be CGC, of which the CG is belonging to the first codon also, that is ACG. So the CGC codes for arginine. Then overlapping again is the next codon forming the alanine, and then the next codon which codes for arginine again. So here what is interesting to note is that if the genetic code were overlapping, you would not have got a dipeptide, you would have got a tetrapeptide. However, what has been observed from a lot of experiments is that it is always a hexanucleotide giving rise to a dipeptide. And so continuous and non-overlapping is the case. So when you say contiguous, what we mean is there is no gap in between any of the codons. This is not there. Neither is any gaps present between any of the nucleotides. It is a continuously read system. So there will be basically what is understandable is if you have an ACG, you have a CGA and an ACG again, it is not like ACG will be read, CGA will not be read, and then an ACG will be read. No, there are no gaps. It is continuously read. So that is the second rule. Now the third rule is, well, before going to the third rule, UAA and UGA are defined codons. Okay. Now let us look at what it defines in the genetic code. Okay. So the third rule is that it represents, or the genetic code has a specific reading frame. Point to note is, suppose you have a 9-mer, so you have a sequence made up of 9 nucleotides, okay, and this is, if it is read from the 5' end of the first nucleotide itself, then you have ACG, ACG, ACG coding for a tripeptide threonine, threonine, threonine. But if it is read from this point, okay, so the reading frame is different. So it's not being read from here, it is read from here. So now the reading frame will go as CGA, 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 continued, okay. So what is going to happen is that now you will not have threonine present in the proteins, but what you will have is arginines as the amino acids. That's because the reading frame has changed, and therefore the codon that is being read is not the one which would have begun from here. Again, if you have a reading frame here, you would have a set of amino acids which is totally different from the first two. That's because the codon is changing. Unless you have a polynucleotide made up of only A. In that case, whether the reading frame is here, here or here, you would still get only the same amino acid. However, the number of amino acids may vary. So the reading frame is decided by the start codon. So you have a codon that is defined to start translation, to start the synthesis of proteins, and that start codon is AUG. This is present in prokaryotes as well as in eukaryotes. In eukaryotes, it is mostly universal, but in some prokaryotes, you may have other codon also that codes for a start codon. So you cannot say that the start codon is universal, but you can definitely say it is nearly universal. And then we come to this, you have the stop codons. So there are 3 codons which are acting as stop codons. Now, when you talk about stop codons, none of these codons are going to code for any amino acid. The first codon to be identified or discovered was the UAG, and it was again an interesting development that happened because of a study that was being carried out for something else. So in a lab in Caltech, okay, the principal scientist, basically Seymour, uh, they were studying bacteriophages, and there was a mutant bacteriophage which had a typical growth pattern. So the principal scientist, Dr. Seymour, announced in the lab that whoever is able to identify the mutation, that mutation will be named after that person. And his student Harris Bernstein, okay, identified the mutation as UAG. And Bernstein in German stands for amber, which is a color. So, therefore, UAG was referred to by amber. When the next codon was identified to be a stop codon, which is UAA, keeping in consideration the color code, the second one was named as ochre. And the third one, UGA, which is the rarest of the stop codons, okay, was named as opal. So that is how you have in the codons a start codon and a stop codons. So any codon between any codons, any number of codons between the start codon and the stop codon will be read by the ribosome and translated into a protein. So therefore there is a reading frame. This reading therefore will always end with the stop codons. Now, therefore from all the previous slides what we have understood is that the codon is a triplet. So how was it identified that the codon is a triplet, or how was it known, or how was it understood that it is a triplet? Well, if you look at the possibilities per se, if you consider that one nucleotide codes for one amino acid, then there is only 4 amino acids possible, which is not really the case because we know that there are 20 amino acids and in fact more now. So therefore, this was completely rejected. So then it was looked at, suppose the codon is a doublet. If it is a doublet, then the number of possibilities is 16. So a doublet codon will code for 16 amino acids, which is again falling short of the 20 amino acids. So therefore, when one goes to triplet being a codon, there are 64 possibilities. Okay, and therefore the 20 amino acids are covered by these 64 codons. But what is interesting to note over here is that now you have extra codons for these amino acids, indicating that there is a possibility that one amino acid can be coded for by more than one codon. And that is actually the case because genetic code is also degenerate. So that the genetic code is a triplet was confirmed by a simple experiment carried out by Francis Crick and Sidney Brenner and their co-workers. So they took a gene again from bacteriophage, T4 bacteriophage, and what they did is they inserted single point mutations and checked what was the result with respect to the reading frame. So they inserted a single insertion. So that was the first mutation that they induced, and they obtained a gene sequence which had the insert present. And what is observed is from the point of the insert, the reading frame got altered. So with insertion, with one nucleotide insertion, there was an alteration in the reading frame. When a second nucleotide was inserted, you have two inserts. So from the point of inserts, again what was observed is that there was an altered reading frame. When the third single point mutation was done with another insertion of a nucleotide, there were 3 inserts, and in this case what was found is that there was a difference in the number of amino acids in the protein, but overall the reading frame was maintained. So one could have 3 amino acids which are different from the original protein, or say 1 amino acid which is more from the original protein, but the overall reading frame did not change. Whereas in these 2 cases, what is observed is from till the point of insertion, the amino acids were same as the original. But after the point of insertion, the amino acids began to vary. So the gene could tolerate 3 insertions, but not 1 or 2 insertions. And therefore, it was indicative of the fact that you could have 3 nucleotides coding for 1 amino acid. And therefore, the reading frame did not change. If it is a singlet or a doublet, the reading frame changes. But if it's a triplet, the reading frame does not change. So this is how they understood that genetic code is definitely a triplet. Now going to the fact that, yes, so therefore this is what the understanding is. Now going to the fact that genetic code is degenerate. So when you look at the entire genetic code table, so 5' end of the genetic code, This is the second nucleotide in the genetic code, and this is the third nucleotide in the genetic code, codons. So there are interesting observations about this genetic code, of which a few we will take up. You can already see that you have three stop codons which are marked red over here, which is the UAA, UAG and the UGA. Now, in the earlier slide, we had referred to the fact that one amino acid could have more than one codon. So, codons that are specific for one amino acid are called as synonyms. So, consider here, there are 4 codons coding for serine. So, all these 4 codons are considered to be synonyms. And you can make out that the degeneracy is here. You have more than one codon coding for one amino acid, 4 for one, and therefore degeneracy. Another important fact that was observed is when the first two nucleotides are the same and the third is different, they can code for a single amino acid. So have a look at over here, CCU, CCC, CCA and CCG. So only the third nucleotide varies, and with that variation, it does not make an obvious change in the amino acid that it codes for. So effectively what we understand is that such changes or alterations or such mutations are what are called as silent mutations. So you can see the difference or you can see the alteration in the DNA, but you will not see the alteration in the protein. So you can see how the genetic code has evolved to minimize the deleterious effect of the mutations. Here the mutation is silent, completely silent. Okay. Now another observation. If you have codons with purines in the second position, then such codons code for polar amino acids. While if the second position is of pyrimidine, then such codons code for hydrophobic amino acids. So let us look at this, where the second nucleotide in the codon is a purine, G, correct? So because it's a purine, you can see that it is coding for serine, which has OH in its as a functional group, and so it is polar. You have arginine with NH2 groups in its side chain, and therefore that also is polar. But consider this set where you have the pyrimidine as a second nucleotide, and all these codons are coding for valine, and valine is a non-polar amino acid. So that is one observation. Now, GCCG with its third nucleotide variable, it codes for the same amino acid. Again, that is interesting. But before we go to this, another fact that I would like to show you is, suppose the second nucleotide is a purine and it is replaced by another purine. So instead of CGU, you have a CAU. Interesting to notice that CGU is coding for arginine and CAU is coding for histidine. So, although there is an alteration from G to A and the amino acid that it is coding is histidine, so there is a change in the protein sequence from arginine to histidine. But take note, both arginine and histidine are basic amino acids. So the change has happened, but the changes again happened in such a way that they are similar in their property, that they are basic. So maybe because they are similar amino acids, there will not be a very big change in the structure of the protein. And so again, the effect of the mutation has been minimized. So coming to this point again, so suppose you have the GC, as we saw, this is all GC, and this is CG, okay. So here what you have is that the third nucleotide is varying, correct? And therefore these 4 and these 4 code for the same amino acid. Please understand that CG will bind to GC in the anticodon of the tRNA. So again it is a GC base pair. GC base pairs have 3 hydrogen bonds. So since they have 3 hydrogen bonds, a total of 6 hydrogen bonds will be formed, which makes the interaction of this with its anticodon strong. So even if the third nucleotide is not binding strongly, there is an allowance. Okay, so therefore you will have CGU, or whether it is CGC, or whether it is CGA, all of them code for the same amino acid. But however, if the first two codons are of A or U, then it is not necessary that, depending on the fact that you have the first two nucleotides as AU, you will have them coding for the same amino acid. So look at over here. You have the UUU, you have the UUC that codes for phenylalanine, but a UUG codes for leucine. So in this case, you do not see that when you have the first two nucleotides as UU or AU, they're not coding for the same amino acid, unlike in this case. So these are some important facts about the genetic code per se. Now going to another interesting aspect of the genetic code, which is the Wobble's hypothesis. So we all know that in an mRNA, when you have a codon, say for example, you have AAA over here, then a tRNA will come and hybridize with this AAA, but to hybridize, it should have complementary base pairs. So the anticodon over here is UUU. Now, suppose the mRNA is read always from 5' to 3', then the anticodon will always be anti-parallel to this codon. So you have the 5' end of the anticodon here and the 3' end of the anticodon here. Please note that this tRNA will always bring an amino acid that is coded by the codon AAA. So say for example, AAA codes for lysine, then this tRNA will be bound to by lysine over here. Okay, so what was proposed by Francis Crick as Wobble's concept is that the nucleotide at the 5' end of the anticodon can wobble. Now what do we understand by that? So this is something that we have already observed happening over here. Now this nucleotide, okay, Francis Crick said that this nucleotide could also interact or form hydrogen bonding with G. So this nucleotide, the same anticodon, could recognize two codons with the variation only in the third codon. The third nucleotide of the codons. So therefore, this U can wobble between A and G. What does this mean? It means that this tRNA is able to recognize two codons. And this is true because what has been observed is that suppose you have 64 codons as observed, okay, and you have 4 codons which are very highly defined. So 3 are stop codons and 1 is a start codon. The remaining codons are 60 codons. So 20 amino acids can have more than one codon. So therefore, if there are 61 codons present coding for amino acids, then there should be 61 tRNA. Each tRNA being able to form hydrogen bond with its respective codon. But what has been observed in reality is that the number of tRNA are much lesser than the number of codons. So therefore, this explains that why the number of tRNA are less, because the same tRNA can actually recognize two codons, and therefore you can already see that if you have to have two tRNA over here, now one tRNA suffices. So this is something that is very important and has been understood by understanding the Wobble's concept. So based on the Wobble's concept, therefore, the pairing is such. Well, when you have in the 5' end nucleotide, always understand it is the 5' end nucleotide of the anticodon. If it is A, it will only always bind to U. So here you do not have any wobble. But if it is U, as we can see, it can interact or hydrogen bond with both A and G, as you can see over here. G can with U and C. C cannot wobble, but there is a fifth nucleotide that is present in tRNA. Please note again that tRNA has several rare nucleotides present in its structure. And one of the rare nucleotides that is present is inosine. Inosine is nothing but a nucleoside attached to hypoxanthine. So the nucleotide that is there, that is inosine, is able to bind to not one or two, but they're able to bind to three nucleotides. And so you can have, if you have tRNA that has I at this position, then that tRNA can recognize three codons. Okay, so that is the understanding. So let us look at the rules of the wobbles concept. So the first rule is that generally the wobbling is always such that you have a purine and a pyrimidine interaction. So the distance between the ribose and the ribose is never compromised on. So you will always have a inosine binding to an adenine or a guanine binding to a uracil. So again you can see it's a purine and a pyrimidine. Again you can see it's a purine and a pyrimidine. So that is not compromised on at all. So that is something that is there. The second is it does not permit one tRNA to bind to more than three codons, not more than three codons, which is if inosine is present, as we have already observed. So let us take into consideration serine. Serine has six codons and there are three tRNA that can recognize the total six codons. So instead of having six tRNA, you can already see that serine just has three tRNA. So you can see why the number of tRNA are much lesser than the number of codons. So in the Wobble concept, the understanding is why is that just that the 5' end nucleotide of the anticodon is the one that is wobbling. So what is clear is that this wobbles, but not the 3' nucleotide. If this is considered to be the anticodon, this portion is the anticodon. When you look at this structure, the tRNA has a very typical structure with an inverted L shape, and this region over here is what is the anticodon arm, and this loop over here is the anticodon loop. Now, a loop will never have the nucleotides base pairing. That is something that is observant. And there are five nucleotides over here that stack with each other, and the five nucleotides includes the anticodon. So this is the five nucleotides that you can observe that stack with each other. So if you consider 3' nucleotide of the anticodon, then it is stacked between the 37th nucleotide and the 35th nucleotide. So this does not have spatial, that is space wise, it does not have much flexibility. But when you look at the 5' end nucleotide of the anticodon, this does not have stacking at this end. Since it has no stacking at this end, this is free to wobble. And hence it is always the 5' nucleotide that is observed to be wobbling between two nucleotides in the codon in the mRNA. So this is something that has been understood and therefore attributing to its being able to wobble. This is what is known. Now let us look at how the code was cracked. Now what do we mean by cracking of the code? So we know that there are 61 codons that code for amino acids. AUG being the start codon, AUG is coding for, so that is known. That is what we know. Now, whether AAA codes for lysine or CCC codes for lysine or or UUU codes for arginine, how do we know that? So which codon is assigned to which amino acid? That is what we understand by cracking of the code. So how was it carried out? Again, by a few very simple experiments. And one of the reasons why these experiments were successful were because in the lab, the RNA templates could be synthesized using an enzyme called polynucleotide phosphorylase. So there were several polynucleotides that were synthesized in the lab, and of which, say for example, something like this, if you use only ATP as the nucleotide and the polynucleotide phosphorylase forms an RNA, then the RNA will have AAAAA, poly A. Similarly, U's. So the first RNA that was synthesized was poly U. Another interesting fact that led to the cracking of the code was that these synthetic RNA templates could be synthesized in a cell-free translation process. What does one mean by cell-free translation? So we all know that translation happens within a bacteria because the ribosome is present and the ribosome carries out, it binds to the mRNA, it allows the tRNA to bind to the mRNA and allows it to synthesize the protein. So all this thing happens inside the bacterial cell. Now, when you lyse open the bacterial cell, what you get is a cell extract. And what was observed is that you could still have protein synthesis in that cell extract. So that is why that is called as a cell-free translation. So what was actually carried out was that E. coli was taken, it was lysed open, and the extract that was obtained was used for translating this poly-U. Okay, so and another interesting thing that was done is that the amino acids that they used for the translation process were labeled, so they could actually find out what amino acid is getting attached. So when this is translated, what was obtained was a polyphenylalanine. Because it is only U. That means that U codes for a phenylalanine. So you see, cracking of the code happened with understanding that a poly U gave rise to polyphenylalanine. There is only one amino acid that was bound to bipeptide bonds to form the polypeptide. So you could have only one codon possible over here, which is U, and that is standing for phenylalanine. Similarly, poly A, when was translated, one obtained polylysine. So what does it tell? This tells that AAA codes for lysine. Poly C was also done and that coded for proline, and so on and so forth. Now, this is using single nucleotides. The next layer was to go one step up, and that was to make RNA using two nucleotides. So to get what is called as copolymers. So say for example, there is a reaction mix in which the number of A was greater than number of C. So there are all possibilities that you will have more of AA and then a C depending on the ratio. So you will have AAC, AAC, AAC. In some points you may have CCA, CCA. So when they observed this after the translation of this copolymer, they found that there was a polypeptide in which the majority of the amino acids are asparagine and few are histidine. So it was therefore deciphered that AAC coded for asparagine while CCA coded for histidine. So that is how again this was cracked. So similarly, many such copolymers were made and eventually all of them could be decoded this way. But there was another approach that was done to actually kind of corroborate with whether this is true or not. So the other approach that was made is to find out which amino acyl tRNA goes and binds to the ribosome when the ribosome is bound to the mRNA. And interestingly, what was done is that it was only a trinucleotide that was synthesized, and that trinucleotide was attached to by the ribosome. And when you have this ribosome trinucleotide complex formed, you would observe which amino acid would come in, because the amino acid has the anticodon. So it can come and bind to this trinucleotide. So looking at which is the amino acid tRNA that comes and binds to the ribosome mRNA complex, it was concluded what does the codon code for. So suppose you have a U, and a phenylalanine tRNA comes and binds to the U, then you know that the UUU codes for phenylalanine. If you have GUU, then the tRNA for valine comes and binds. So therefore you can decipher that GUU codes for valine. So this was another approach. Now, genetic code, as mentioned earlier, is nearly universal. It is not completely universal, but nearly universal. Why is it nearly universal? Because what has been found is that in several subcellular organelles, like in our mitochondria or in plants chloroplast itself, it has been found that many of the codes were found to be different. So UGA, a stop codon for the genomic DNA, is not a stop codon in the mitochondria. It rather codes for a tryptophan. So you can see that there is a difference. So you cannot say that it is universal, right? AUG and AUA both code for internal methionine, right? Whereas Otherwise, it is AUG which codes for methionine alone. In mammalian mitochondria, another observation, AGA and AAG code for arginine in genomic DNA, but in the mammalian mitochondria, they code for stop codons. So again, a variation. In certain bacteria too, there are deviations from the standard codes. So say for example, Mycoplasma, UGA, which is normally a stop codon, is coded for tryptophan. So therefore you see that difference. Plus what has been found is that several bacteria show something that is called as codon bias. So when a bacteria needs a certain amino acid to be incorporated more in their proteins, then what they do is several of the codons are used to code for the same amino acid. So there is a bias towards a particular amino acid. So many of the codons, say for example, you have one codon coding for lysine, but then because the arginine is being added more in the proteins in the bacteria, then a codon for lysine can code for arginine in that bacteria. So that is what is called as codon bias. So with this, let us conclude. The codons are read from 5' to 3' continuously without any overlaps and within a reading frame. The codons are triplets and they are degenerate. The Wobble concept proposed by Francis Crick allows a tRNA to bind to more than one codon, as the 5' end nucleotide of the anticodon can wobble between binding to two nucleotides. And there is presence of inosine in the anticodon, which is a rare nucleotide, and it has been found to bind to three nucleotides. So wobbling between three nucleotides is also possible. The genetic code definitely has a start codon and it has three stop codons, and all the other 61 codons, rather 60 codons, because one of them is already a start codon, and the other 60 codons code together for all the amino acids, which are nearly universal, with exceptions found in several bacteria and subcellular organelles. Thus, the blueprint in the DNA coding for the structural and functional molecules in a cell is encoded in the form of genetic code, a rule book that allows systematic deciphering to form a polypeptide. Thank you.